Okay, so we're going to start chapter 13 today, which is meiosis in the sexual life cycle. Somebody want to hit a couple of those middle buttons and turn down the lights a little bit? It'll be easier for you to see it. Yeah, you just keep pushing until something goes off. There does not appear to be any rhyme or reason to how it works. Thank you. So in this section here, we're looking at the sexual life cycle, which is different than mitosis. With meiosis is where you're looking at passing traits from one generation to the next. With mitosis, you are just making clones of the previous generation in there. So this will actually involve heredity, which is the transmission of traits from one generation to the next. And genetics is just the study of heredity and how you get this hereditary variation in there. So your offspring get their genes from their parents by inheriting the chromosomes. So the genes are the units of heredity that are going to program for specific traits. Your gametes are going to be the sex cells or reproductive cells in your plants and animals that are going to transmit the genes from one generation to the next. So it's your sperm and egg. The somatic cells are all of your body cells except the gametes in there. In human cells, we have 46 chromosomes. When we talk about where specifically a gene is located on a chromosome, it's the locus, or loci if it's plural in there. So when you compare asexual and sexual reproduction, asexual reproduction is just going to make a genetic copy of themselves. A single parent is going to be the sole parent in there. So it doesn't involve more than one cell, it's just one giving rise to two. And the clones are the genetically identical individuals that arise from that parent. With sexual reproduction, you're going to give rise to offspring that are going to have their own unique combinations of genes from the two parents in there. So when we talk about the life cycle, it's a stage of an organism from conception till it makes its own offspring. In terms of life cycle for biology, we don't care what you do after you make the offspring in there. That's what we're looking about, is from when you start till you make an offspring. That's the life cycle. When we look at the chromosomes, one way of looking at them is a karyotype, which is essentially a photograph of the chromosomes where they get arranged into pairs starting with the longest chromosomes, and then they're put in order in terms of their size in there. So when you take the picture, it doesn't look like that. You have to take the picture, cut out the chromosomes, and arrange them like that. So there are websites that have places where you can go on and practice organizing them like this. There really isn't anything you would need to do that for. It's just so you can see that they are in that order. You can see starting with number 1, those are the longest. When you get down to 22, these are the smallest. The 23rd. You'll notice here there's 2 different ones on there, X and the Y. The Y is significantly smaller than the X chromosome in there. So the homologous chromosomes are the 2 chromosomes that are going to make a pair. They're going to be the same length, have the same centromere position, and they're going to stay the same. Your sex chromosome is going to be the one that determines the sex of the individual in there. XX is female, XY is male. Only a small part of that is homologous in there. You can't have the whole thing be homologous when you don't have all of the same parts in there. So your autosomes are your non-sex chromosomes. So a cell that's diploid is one that's going to have 2 copies of the chromosomes in there. It's abbreviated as 2N. So all of your cells except your gametes are going to be 2N or diploid as a human, and in most animals. The haploid cells are going to be your gametes. Those are going to contain just a single set of the chromosomes because they're going to fuse with somebody else's set of chromosomes. So in order for that to fuse, you'll have fertilization that will occur. That's just the union of the 2 nuclei in there into one cell. And what it makes then is the zygote, is the fertilized egg. And meiosis is going to be the process by which you make these sex cells in there. So between plants and animals and then the fungi and protists in there, we do have some variety in the sexual life cycles in there. And this diagram actually explains it pretty well in there. So in animals, you can see they're over on the farthest side over there. Just scoot it up just a tiny bit so you can see that's animals in there. So animals spend most of their life in the diploid state in there. You can see the mouse at the bottom. The multicellular organism is the diploid one in there, and that's the dominant phase in there. So after you have fertilization, you become diploid in there, and you spend most of your life there. The only time you have the haploid state is just the single-celled gametes in there. So that's just a small portion of your life cycle when you are the separate sperm and egg, and you really aren't even you yet at that point. In plants, it's a little bit different. You can actually have two versions of the plant even occur in this. So they have the haploid stage in there where you go from the spores and then they make the gametes in there. And then when you have fertilization occur, you can go in and have a multicellular diploid state as well. So sometimes you'll see it being referred to as having the gametophyte generation and the sporophyte generation with those. And then with the fungi and the protists, they will tend to be multicellular or unicellular, but dominant in the with the you're going to have in here your gametes are going to be the majority of the life cycle, and then the zygote is basically only formed in there long enough to do meiosis and split into the haploid cells again. So there's just the difference in how much time you spend in the different phases in there with the dominant part of the life cycle in there. Plants are the ones that are right in the middle, and different types of plants will actually have more dominant phases in there. So some plants are sporophyte dominant, some are gametophyte dominant. So meiosis is going to reduce the number of chromosome sets. So to go from being diploid to haploid, and you've got two phases to meiosis, meiosis 1 and 2, and it ends up resulting in 4 daughter cells. So here you've got interphase in here. This is a pair of homologous chromosomes, so even though they're two different colors that it uses for demonstration purposes here, it's the two legs of one chromosome that you're seeing there. So they've separated here so they can make a copy. And with the two copies, you end up with sister chromatids on there. When it undergoes meiosis 1, those are going to split and go into 2 different cells in there. And then they will split again in meiosis 2, and you get 4 different cells in there. So you can see where these sister chromatids have separated in there. So you have the homologous chromosomes separate, and then the sister chromatids separate in there. So the chromatid is that individual leg. The chromosome is the whole big thing. And if that helps you to remember, you have to split the whole big thing and then you split the little thing first in there. So when you compare meiosis and mitosis, if you look in here with the parent cell, you're going to have chromosome duplication that goes on at the beginning in there. And you've got the chiasma here where you've got the 2 chromosomes that are duplicated here for the homologous chromosome pair. Metaphase, everything lines up, except here you've got the homolog pairs lined up. Here you just have the individual chromosomes lined up. In anaphase and telophase 1, things are going to separate, and you have the 2 daughter cells here. And then you go through meiosis 2, and you're going to split up the genetic information in those daughter cells to make 4 daughter cells that are all haploid there. So there's 3 events that are unique to meiosis in there. one of the things you're going to have is synapses and crossing over. So synapses is going to be where you have the duplicate homologous pair and they form this synaptonemal complex. So that's where you have the two chromosome homologs come together, and that synaptonemal complex is just where they attach together in there. Crossing over is going to be where you have the genetic information that are on those homologous pairs switch around on different chromosomes. So when you make a sperm and an egg, you're going to send one of your chromosomes, one of each of your chromosomes, into that sperm or egg, whichever you make. However, it's not going to be an exact copy of your chromosome in there. It's going to be a copy that's a unique mix of the two copies of the chromosomes you have. So if you look at it like this, instead of having so what you have is this is your unique pattern in there. When you have crossing over, what's going to happen is you're going to have part of these trade places. So what you end up making is this. So you'll pass one of these on in there when you do crossing over. So crossing over just means chunks of the same region cross over to the other chromosome in there to make a new combination in there. So you'll have the homologous pairs line up at the middle in the metaphase plate rather than just the individuals in there. And then you'll have separation of the homologs in there. But the sister chromatids stay together the first time and they go apart the second time in there in anaphase 2. So the chiasmata is going to be what holds the homologs together as the spindle forms for the first mitotic division in there. So one division is what we call reductional, the other one's equatorial in there. During meiosis 1 you have reductional where it's going to have the number of chromosome sets per cell. And then in meiosis 2 you're going to have the sister chromatids separate. So one of the big things with sexual life cycles is it gives a lot more genetic variation in the offspring in there. And the behavior of the chromosomes during meiosis and fertilization is going to be what gives you a lot of that variation in there. So one of the things you'll have is independent assortment of the chromosomes. So during metaphase 1, you're going to have one maternal and one paternal chromosome on there. And so here you can see you have two of the blue lined up on the same side, the two red on the other side. It is just as equal probability that you will have one blue, one red, one blue on there. It's a 50-50 chance of are you going to be on the left or the right in there, or the top or the bottom of it in there. And so you've got two possibilities for every chromosome. And you've got 23 different chromosomes. So when you look at the number of possible combinations of chromosomes that you can pass on to your sperm or egg, it's 2 to the 23rd, or 8.4 possible million combinations of the chromosomes. So if you really like your first child and you're thinking, I want to make another one just like it, probably not in there. That would require making a whole lot of babies to have the chance that you would make the same egg again. So then when you throw in crossing over in there, you're going to get even more variation because typically you're going to have 1 to 3 crossover events per chromosome in there. So that would take it from being 5 to the 23rd to, or 2 to the 23rd to possibly 3, 4, or 5 to the 23rd in there, possible combinations in there. So this is just showing here where you've got the non-sister chromatids, where you form the chiasma. The centromere is here. You've got some crossing over going on with anaphase 1. When you separate them in anaphase 2, now you can see that there is the different combinations in there, and you end up with 4 different daughters in there that are going to have recombinant chromosomes. So then with random fertilization in there, so in each human, male and female, your gamete represents about 1 in 8.4 million possible combinations in there. So then when you fuse a gamete from a male and a female, you multiply those together, and that gives you 70 trillion possible combinations. So if you are looking for getting the same child with the same spouse or the same partner in there, 70 trillion tries to get it a duplicate in there. And it's still not going to be a duplicate in there. So what has this genetic variation done for us within the population? Natural selection results in an accumulation of the genetic variations that are favored by the environment. So the genetic variation that is going to be better suited to dealing with the environment, the idea is if you're better suited to deal with the environment, you're more likely to get a mate, you're more likely to reproduce a child and survive. If your traits are poorly adapted for the environment, you probably won't get a mate and reproduce. So mutation is going to produce different alleles along with sexual reproduction in there. So that's going to give you even more genetic diversity in there. So what you'll see is asexual reproduction is more advantageous in stable environments. It's assuming nature's figured out which genes it works, and it will just perpetuate those because that's a successful combination. If the environment is less stable, you're better off to have sexual reproduction. Sexual reproduction is almost universal among animals, except there's one exception, there's rotifer in there. You're probably not going to get the animals to say, you know what, I no longer want to sexually reproduce. I think most animals kind of like it, or at least the practice part before the baby comes in there. So it's where they say the asexual reproduction more advantageous in a stable environment. We don't really have the option to choose which kind of reproduction we're going to do because we decide our world is unstable. So horizontal gene transfer. This can occur in bacteria, but also in these little rotifers in there. So the little rotifers, they don't get to have sex in there. And so instead, when a rotifer needs to get some genetic variation, what happens to create that variation in their population is they dry up and crack, and then their DNA falls out into the environment. And when you're drying up and getting cracks, you can pick up DNA from the environment and take it in. So instead of sex, they basically get to crack and pick up junk from the environment to have their genetic variation in there. So be good or I'll turn you into a rotifer. So we'll stop there today. That's chapter 13 in there. So Friday after the test, we'll start on 14 with Mendel and the inheritance in there.